Welcome, curious minds, to the deep dive. Just look around you for a second. That tree outside, the salad you had for lunch, maybe even just the air you're breathing right now.
Hmm.
Almost everything living relies on this one process. But how? How does plain old sunlight build a tree trunk or, feed basically everything?
It's an amazing question, isn't it? And the answer is photosynthesis.
Okay.
That's what we're diving into today.
Okay.
We're going to sort of peel back the layers, go step by step into how plants, algae, some bacteria,
Uh-huh.
how they turn solar energy into food energy.
Sounds good. So where do we start? To really get it, we need the context, right? The big picture of life.
Exactly. It really starts with how organisms get their food, their energy. You've basically got two camps, autotrophs and heterotrophs.
Autotrophs. Auto meaning self, right. Self-feeders.
So exactly. They're the producers. They make their own complex organic stuff like sugars from really simple inorganic things, CO2, water.
And the ones we're focused on use light. Photoautotrophs.
That's the main group, yeah. Plants, algae, cyanobacteria. They use sunlight to power this whole synthesis. They're the foundation.
And then there's, well, us. And most other things. Heterotrophs.
Right. The other feeders. We can't make our own food from scratch like that. We need to consume organic compounds that others have produced.
So eating plants or eating animals that ate plants.
Or even decomposers.
Right.
Like fungi breaking down dead leaves.
Mm.
It all traces back. And here's the kicker. Almost all of us heterotrophs depend completely on those photoautotrophs. Not just for food, but critically for the oxygen we breathe. That's a byproduct of their process.
Wow. Okay, yeah, that dependence is huge when you think about it. So this photosynthetic magic, where does it actually happen in a plant? Is it just like all the green bits?
Well, yes and no. It happens in specific little factories inside the cells called chloroplasts.
Okay.
And while all green parts have them, the leaves are the absolute powerhouses. Get this. About 500,000 chloroplasts per square millimeter of leaf surface.
500,000. That's incredible density.
It really is.
And how do the raw materials get in? The CO2 and water.
Okay, so CO2 from the air enters through tiny pores on the leaf called stomata. Think of them like little gates. And oxygen gets out the same way.
Stomata. Got it. And water.
Water comes up from the roots, delivered through the veins in the leaf. Those veins also act like plumbing to export the sugar that's made.
Right, the transport system. Okay, so let's zoom in. If we could shrink down and go inside a chloroplast, what's the layout? Paint us a picture.
All right, imagine a tiny bean shape wrapped in two outer membranes. Inside that is a thick fluid, kind of a soup, called the stroma.
The stroma.
And floating within that stroma is another complex membrane system. It's made of flattened sacs called thylakoids.
Thylakoids,
What?
like little pouches.
Exactly. And these thylakoids are often stacked up like piles of pancakes or coins. Each stack is called a granum.
A grano. Okay.
And here's the crucial part. The chlorophyll, that green pigment that captures the light. It's embedded right in the membranes of those thylakoids. That's where the light capture happens.
Wow, okay. So the structure itself is perfectly designed for this. Now, photosynthesis is really complex, but there's a basic chemical equation people often see.
Yeah, the simplified one is 6CO2 plus 6H2O plus light energy gives you C6H12O6. That's glucose, a sugar, plus 6O2.
Carbon dioxide plus water plus light makes sugar and oxygen. Seems simple enough on paper.
Right, but that summarizes a huge number of individual steps, and actually the direct product isn't glucose itself, but a smaller 3-carbon sugar that the plant then uses to build glucose and other things.
Ah, okay. And one of the big historical questions was where that oxygen actually comes from. It wasn't obvious.
Not at all. For a long time, scientists thought plants split the CO2 and maybe added water to the carbon, so the oxygen would come from the CO2.
Makes sense intuitively, I guess.
But then in the 1930s, a researcher named C.B. Van Niel was studying these interesting sulfur bacteria. They did a kind of photosynthesis, but used hydrogen sulfide, H2S, instead of water, H2O.
And what did they release?
Sulfur, not oxygen. So Van Niel had this brilliant insight. He proposed that maybe all photosynthetic organisms need a hydrogen source, and they split that source. Sulfur bacteria split H2S, release sulfur, so maybe plants split H2O and release O2.
That's clever reasoning. How did they prove it?
Much later, with isotopic tracers, they used a heavy form of oxygen, oxygen-18. They could label either the water or the CO2 given to the plant, and they found the O2 released only contained the heavy oxygen when the water was labeled.
Ah, definitive proof. The oxygen comes from splitting water.
Exactly, a landmark discovery.
So thinking about it chemically, photosynthesis is a redox process, right? Like cellular respiration, but backwards.
In a way, yes. Redox means reduction to oxidation, the transfer of electrons. In photosynthesis, water gets oxidized, it loses electrons and hydrogen ions.
Okay.
And CO2 gets reduced. It gains those electrons and hydrogens to become sugar. But here's the key difference from respiration. Energy. Respiration releases energy as electrons fall down, an energy gradient. Photosynthesis requires energy input to push those electrons uphill from water to sugar. It's energonic.
And that energy input
Is light. That's the crucial solar power boost.
Okay, this makes sense. So this whole complex process, you mentioned it happens in stages.
Yes, two main stages that work together. First are the light reactions. You can call them the photo part.
Capturing the light.
Right. They happen in those thylakoid membranes we talked about. This is where solar energy is converted into chemical energy. Water gets split here, releasing the O2, and the captured energy is stored temporarily in two key molecules, ATP.
The cell's energy currency.
Exactly. And NADPH, which is basically a high-energy electron shuttle.
Okay, ATP and NADPH generated. What's the second stage?
That's the Calvin cycle. This is the synthesis part. It happens out in the stroma, the fluid part.
And it uses the stuff made in the light reactions.
Precisely. The Calvin cycle takes the ATP and NADPH from the light reactions and uses their energy and electrons to take CO2 from the air and build sugar from it.
So the Calvin cycle itself doesn't need light directly.
Correct. But it completely depends on the light reactions finishing their job first to provide the power. So usually both run during the day. They're tightly linked.
Two interconnected phases. Let's dive deeper into those light reactions then, starting with sunlight itself. What is light, really?
Good question. Light is a form of electromagnetic energy. It travels in waves. What matters for photosynthesis is the visible light spectrum, the rainbow colors, basically from about 380 nanometers violet to 750 nanometers red.
Nanometers, that's the wavelength.
Yes, the distance between the crests of the waves. And light also behaves like it's made of little packets of energy called photons.
Photons.
And here's a key point. The shorter the wavelength, the more energy each photon carries. So violet light photons have more energy than red light photons.
Okay. And how do plants actually catch these photons?
With pigments. Pigments are just molecules that absorb specific wavelengths of visible light.
Like chlorophyll. That's why leaves look green.
Exactly. Chlorophyll is brilliant at absorbing violet blue light and red light, but it doesn't absorb green light well. It mostly reflects or transmits it. So that's the color we see.
Clever. Are there other pigments involved besides chlorophyll?
Oh, yes. There's chlorophyll A, which is the main key player in the light reactions. But there's also chlorophyll B and another group called carotenoids. They look yellow or orange.
What do they do?
They're accessory pigments. They can absorb light at slightly different wavelengths that chlorophyll emisses, broadening the range of light the plant can use. Think of it like extending the reach of the solar panel.
Ah, okay. More coverage.
And carotenoids have another crucial job: photoprotection. They help dissipate excessive light energy that could otherwise damage the chlorophyll, like sunscreen for the cell.
Sunscreen, I like that. So what happens right at the moment a chlorophyll molecule absorbs a photon?
Okay, so when a photon hits a pigment molecule, its energy bumps one of the pigment's electrons up to a higher energy level, an excited state.
More energy, but unstable.
Very unstable. If that chlorophyll molecule were just floating alone in a test tube, the electron would quickly fall back down, releasing that extra energy as heat and maybe a little bit of light fluorescence. You can actually see this red glow from isolated chlorophyll.
That's not what happens in the leaf.
No way.
Yeah.
In the thylakoid membrane, the chlorophyll isn't alone. It's organized with proteins and other pigments into amazing complexes called photosystems.
Photosystems, like energy harvesting units.
Exactly. Think of each photosystem like a satellite dish. It has a central core called the reaction center complex, and surrounding it are lots of light harvesting complexes.
Okay.
These light harvesting complexes are packed with chlorophyll and carotenoids. They act like antennas. When any pigment in the antenna absorbs a photon, the energy, not the electron itself, just the energy gets passed from molecule to molecule like a wave in a stadium crowd until it reaches the reaction center complex. This complex holds a special pair of chlorophyll molecules. When the energy arrives there, these chlorophylls get excited and actually pass one of their high-energy electrons off to another molecule called the primary electron acceptor.
Ah, so that's the key transfer. Light energy becomes chemical potential energy in that electron.
That's the conversion point. It's a redox reaction. The chlorophyll gets oxidized, the acceptor gets reduced.
And you mentioned there are two types of these photosystems.
Yes, named in the order they were discovered, not the order they function. There's photosystem 2, PS2, whose special chlorophyll pair is called P680 because it's best at absorbing light with a wavelength of 680 nanometers.
Okay, PS2, P680.
And then there's photosystem I, PSI, with a chlorophyll pair called P700, best at 700 nanometers. And crucially, in the main pathway, PS2 acts first.
PS2 first, then PSI. Got it. Okay, let's trace that electron flow. The main pathway is called linear electron flow.
Yes. It's an amazing journey. Step 1. A photon strikes a pigment in a light-harvesting complex of PS2. The energy gets relayed to the P680 chlorophyll pair.
Exciting an electron in P680.
Right. That electron gets boosted and immediately captured by the primary electron acceptor of PS2. Now, P680 is missing an electron. It has an electron hole. It becomes P680 plus sectin.
And it needs an electron back desperately. Where does it get it?
This is where water comes in again. An enzyme right next to PS2 splits a water molecule. H2O becomes 2 electrons, 2 hydrogen ions, H+, and an oxygen atom.
Ah, the water splitting.
Those 2 electrons fill the hole in P680+, returning it to P680. The H+ ions are released into the thylakoid space inside the thylakoid sac, and the oxygen atom immediately combines with another oxygen atom from another split water molecule to form O2 gas. That's the oxygen released.
Wow. So P680 plus is so hungry for electrons it can actually rip them from water.
It's one of the strongest biological oxidizing agents known. Okay, so now the electron captured by PST's primary acceptor doesn't stay there. It gets passed down an electron transport chain.
Like a series of steps.
Exactly. A series of protein complexes embedded in the thylakoid membrane. As the electron moves from one carrier to the next, going from PS2 towards PSI, it loses a bit of energy at each step.
And what happens to that released energy?
It's used to do work. Specifically, it powers the pumping of more H+ ions from the stroma into the thylakoid space. This builds up that proton gradient we talked about, a higher concentration of H+ inside the thylakoid than outside in the stroma.
Storing potential energy in that gradient. And that gradient drives
ATP synthesis. Through chemiosmosis, there's an enzyme called ATP synthase, also in the membrane. It acts like a tiny molecular turbine. As the H+ ions flow back out of the thylakoid space down their concentration gradient through ATP synthase, the enzyme spins and uses that energy to stick a phosphate onto ADP, making ATP. This specific process in chloroplasts is called photophosphorylation.
photo because light started it all. Phosphorylation because phosphate is added. Makes sense. So where's the electron now?
It has reached the bottom of the first electron transport chain and is passed to photosystem I, specifically filling the electron hole in P700 plus space.
P700 plaque. How did it get a hole?
Because light has also been hitting PSI. Photons absorbed by PSI's antenna pigments funnel energy to P700, exciting one of its electrons. This electron gets captured by PSI's own primary electron acceptor, leaving P700 electron deficient, P700 plus. The electron arriving from PS2 fills this hole.
Okay, so light excites electrons in both photosystems. What happens to the electron that left PSI?
It goes down a second, shorter electron transport chain. Doesn't pump protons this time.
Okay, where does it end up?
It eventually gets passed to an enzyme called NADP plus reductase. This enzyme takes 2 electrons from the transport chain and transfers them, along with an H+ ion from the stroma, onto a molecule called NADP plus Li.
Reducing NADP plus Li.
To NADPH, that high-energy electron carrier we need for the Calvin cycle. So at the end of linear electron flow, we've split water, released O2, made ATP via the proton gradient, and stored high-energy electrons in NADPH.
That is incredibly elegant. Complex, but elegant. And you mentioned the chemiosmosis part is similar to mitochondria.
The core mechanism, yes. Both use an electron transport chain to pump protons, create a gradient, proton motive force, and then use ATP synthase to harness that gradient to make ATP. It's a fundamental energy coupling mechanism in biology.
But key differences remain.
Absolutely. Source of electrons? Light-energized water in chloroplasts versus high-energy electrons from food molecules in mitochondria. And the location. Chloroplasts pump H+ into the thylakoid space, making ATP in the stroma. Mitochondria pump H+ out to the inner membrane space, making ATP in the matrix. The resulting pH difference in chloroplasts is huge. The thylakoid space can get down to pH 5, while the stroma is around pH 8.
Wow. Okay, so light reaction's done. We have ATP and NADPH ready in the stroma. Now for the Calvin cycle, the sugar factory.
Exactly. The Calvin cycle is where the synthesis happens. It's anabolic. It builds things up. Like the citric acid cycle in respiration, it's a cycle. It regenerates its starting molecule.
And its goal is to make?
To take CO2 from the atmosphere and fix it into organic molecules, ultimately producing that 3-carbon sugar, glyceraldehyde 3-phosphate, G3P. It uses the ATP for energy and the NADPH for reducing power, both supplied by the light reactions. For every 3 CO2 molecules that enter, 1 net G3P molecule comes out.
Okay, let's track those 3 CO2s. What are the phases?
3 main phases. Phase 1, carbon fixation. This is where CO2 enters the cycle. Each CO2 molecule is attached to a 5-carbon sugar that's already present in the stroma, called ribulose bisphosphate, RuBP.
Attaching CO2 to RuBP, what does that?
The enzyme Rubisco. Ribulose bisphosphate carboxylase oxygenase. This is the enzyme that does the initial CO2 capture.
Rubisco, is that the really abundant protein you mentioned?
That's the one. Possibly the most abundant protein on Earth, given how much photosynthesis happens. So Rubisco attaches CO2 to RuBP, forming a very unstable 6-carbon intermediate.
Which breaks down.
Immediately splits in half, yielding 2 molecules of a 3-carbon compound called 3-phosphoglycerate. So for 3 CO2s entering, we get 6 molecules of 3-phosphoglycerate.
Okay, phase 1 done. Carbon is fixed. What's phase 2?
Phase 2, reduction. Now we use the energy captured in the light reactions. Each molecule of 3-phosphoglycerate gets a phosphate group added from ATP. Then it gets reduced by electrons donated from NADPH.
Using both ATP and NADPH.
Yes. This transforms the molecule into G3P, that key 3-carbon sugar. So our 6 molecules of 3-phosphoglycerate become 6 molecules of G3P.
6 G3Ps made. But you said only 1 G3P is the net output.
Right, because phase 3 is regeneration of the CO2 acceptor, RuBP. Out of those 6 G3P molecules made, only 1 exits the cycle as net product for the plant to use.
What happens to the other 5?
The other 5 G3P molecules, which contain a total of 15 carbons, go through a complex series of rearrangements. This reshuffling uses more ATP and ultimately regenerates the 3 molecules of the 5-carbon starter RuBP that we began with.
Ah, so the cycle can continue. Clever. It uses 5 G3Ps to remake the 3 RuBPs needed to fix the next 3 CO2s.
Exactly. The cycle turns, ready for more CO2.
So let's tally the cost. For 1 net G3P molecule produced, how much ATP and NADPH does the Calvin cycle burn through?
For each G3P synthesized, the cycle consumes 9 molecules of ADP and 6 molecules of NADPH, which of course are regenerated by the light reactions happening simultaneously in the thylakoids.
A continuous supply loop. And that G3P that leaves the cycle,
Yeah.
what does the plant do with it?
It's incredibly versatile. G3P is the starting point. 2 G3Ps can be combined to make glucose or fructose. Glucose can be linked to make sucrose, the sugar transported throughout the plant, or linked into long chains to make starch for energy storage.
In roots or seeds.
Right. Or linked differently to make cellulose, the main component of cell walls' structural material. G3P is the fundamental building block.
It really is the foundation for almost all the organic matter in the plant. Now, plants face challenges, right? Especially heat and drought. Closing stomata saves water but cuts off CO2.
That's a huge trade-off and leads to a problem for many plants called photorespiration.
We mentioned Rubisco, that key enzyme. It can make a mistake.
Yes. When CO2 levels inside the leaf drop low and O2 levels build up, because stomata are closed but light reactions keep making O2, Rubisco can actually grab O2 instead of CO2 and add it to RuBP.
What happens then?
It starts a wasteful pathway. Photorespiration consumes O2, breaks down organic molecules, actually uses ATP, but it produces no sugar. It releases CO2. It basically undoes some of the work of photosynthesis. It can significantly reduce crop yields, for instance.
Seems like a flaw, an evolutionary leftover.
Possibly. It might be baggage from when the atmosphere had much less O2 and much more CO2, so Rubisco's lack of perfect specificity wasn't such an issue. But for plants today, especially in hot, dry places, it's a real problem.
Yeah.
These are called C3 plants because the first organic product of carbon fixation is that 3-carbon compound, 3-phosphoglycerate. Most plants are C3.
But some plants have evolved workarounds, like C4 plants, corn, sugarcane.
Exactly. C4 plants have evolved a clever spatial separation of steps. They have 2 types of photosynthetic cells. In the outer cells, methyphyl, they use a different enzyme, first not Rubisco, to initially fix CO2. This enzyme has a super high affinity for CO2, even when levels are low, and it doesn't bind O2.
So it grabs CO2 efficiently. What does it make?
It incorporates CO2 into a 4-carbon compound, hence the name C4. This 4-carbon compound then acts like a shuttle. It moves into deeper cells called bundle sheath cells, which surround the leaf veins.
Okay.
Inside the bundle sheath cells, the four-carbon compound releases the CO2 again. This creates a very high concentration of CO2 right where Rubisco and the Calvin cycle are located in those cells.
Ah, so it pumps CO2 to Rubisco, overwhelming it so it doesn't grab O2.
Precisely. It effectively eliminates photorespiration and allows the Calvin cycle to run efficiently even when stomata are partially closed.
Very smart. What about cam plants? Cacti, pineapples,
Yeah.
they do something different.
Cam plants, crassulation acid metabolism, use a temporal separation in time. They live in very arid conditions.
So they really need to keep stomata closed during the hot day.
Absolutely. So they do the opposite of most plants. They open their stomata only at night.
At night. But there's no light for the light reactions.
Right. But they can still take up CO2. During the night, they fix CO2 into various organic acids using an enzyme similar to the C4 pathway, and they store these acids in their cell vacuoles. They basically bank the CO2 overnight.
Okay, stored CO2? Then what?
Then, during the day, they close their stomata tight to conserve water. But now, inside the cells, they release the CO2 from those stored organic acids. And now, the light reactions are running, providing ATP and NADPH.
So the released CO2 can enter the Calvin cycle right there in the same cell?
Exactly. They separate the initial CO2 capture night from the Calvin cycle day. Another ingenious way to deal with hot, dry environments.
So C3, C4, CM, all use the Calvin cycle ultimately. Just different strategies for getting CO2 delivered efficiently. That's fascinating adaptation.
It really shows the power of evolution in optimizing this crucial process for different conditions.
And when we step back, all this incredible biochemistry, what's the bottom line? Life depends on photosynthesis. It's more than just sugar.
Oh, absolutely. That G3P is the starting point for everything. About half the organic material a plant makes is actually used by the plant itself for its own cellular respiration, powering its own growth, repair, transport.
Right, plants respire too.
They do. The rest is used for building blocks, making sucrose to send energy to roots or fruits, making cellulose for strong cell walls, think wood, cotton, making starch to store energy for later, like in a potato or a grain of rice.
It fuels and builds the entire plant body.
And on a planetary scale,
Yeah.
it's everything. All the oxygen we breathe came from photosynthesis over billions of years. Scientists estimate plants produce something like 150 billion metric tons of carbohydrates each year.
That number is just staggering. It fuels almost every food web on Earth.
Pretty much. It provides the energy and the organic carbon that nearly all life forms ultimately rely on.
So, thinking about all this, the elegance of the light reactions, the Calvin cycle, the amazing adaptations like C4 and CAM, what's a final thought for our listeners? Knowing how fundamental this is and how cleverly plants manage it, what does it suggest for us facing challenges like climate change or food security?
That's a great question to ponder. I mean, when you see the efficiency, the resilience, the sheer scale of natural photosynthesis, it's humbling, but it's also inspiring. Can we learn from these processes? Can we mimic parts of them for, say, better carbon capture? Or engineer crops to be more efficient or drought-resistant using insights from C4 or CAM pathways? Understanding photosynthesis isn't just biology homework. It's potentially key to tackling some of our biggest global challenges. The blueprint is there, written in chlorophyll and enzymes.
A blueprint for survival and maybe for innovation. Fantastic. Thank you so much for taking us through that intricate world.
My pleasure. It's always fascinating to talk about the process that powers our planet.
We hope this deep dive helps you see the world and maybe even your next meal a little differently. Thanks for joining us. Until next time, keep diving deep.